I am not going to send you the links for videos. You have the video, the current video you have in Teams, and also there is some records from the last year in Moodle. So you have now pretty much chances to see the presentation, the lectures again. So that's easier. Before I start to teach, I would like to give you homework for next time, because what is important for me that you are not only listening. So if you listen only, it's very passive type of learning, passive type of education, and you cannot get pretty much information and pretty much knowledge. So therefore, I would like to ask you to prepare a short presentation to these topics. The presentation should be done next lecture, and you are going to work in groups. So the group will be around 3 students together. And here is the choose. This is the topic which I offer to you to have a presentation, because this is a course of biology. I guess it's pretty fine that you can select one of the organelle and prepare a short presentation. So every student is going to speak 2 minutes, 2 minutes. So it means that you are going to prepare one slide, not more. And together, if you are going in groups of 3, your presentation will be 6 minutes. So this is the topics: endoplasmatic reticulum smooth or roofs, endoplasmatic reticulum, nucleus, plasma membrane, mitochondria, Golgi apparatus, and ribosomes. So please, the most interesting organelle for you, and here write your name. Yeah, for example, here it's some raven, anonymous raven. So just write here your name, and here. So, but be careful. If you write here your name, you have to be sure that also the other students are able to cooperate with you. So it's even better if you just speak together before you book your topic. And you are going to present next Friday. So questions, is it clear?
Are presenting alone or like in a group?
In a group, you are working in a group. So you have to prepare the presentation also in a group. So you are giving a lecture for the topic. So the information are not going to repeat. You have to dissipate the information. So you will find the information, for example, about ribosomes, and you divide it by 3. So everyone from the group will tell a piece, a part of the topic.
Okay.
Is it clear?
Where is the link for the biology 22?
In email.
All right. Thank you so much.
You're welcome. So now let's continue with last week lecture. I think we have finished with water, right? With this slide. I hope so. So we can continue with another one. Could you see the presentation, please?
Excuse me, miss. Can I represent the nucleus?
Right, but you have to talk with your colleagues.
There's already 2 students.
Just ask them if they are able to accept you to their group.
Yes, I already talked with one of them, and I think the other one will accept me.
That's fine. That's perfect. So
Thank you.
let's move to cell structure. This is lecture number 2. And I will give you a short, very brief overview of all organelles in the cell. But it's up to you to make the information deeper. Yeah, so now you will gain some rough information, and then I hope that you are going to develop them. So cell structure. We have 2 types of cell structures. We have 2 types of organisms, prokaryotic organisms and eukaryotic organisms. So therefore, we are going to describe the prokaryotic cell and eukaryotic cell. Typically, for prokaryotic cell, we have, so this type of the cell, we can find it in lower organisms. It's single cell organism, usually. They are evolutionally older, and it is expected that they were here at the beginning. Also, the structure of their cell, it's much simple than the structure of eukaryotic cell. For example, humans, higher plants, all the multicellular organisms, we have the eukaryotic cell. If we look at this picture, we can, here you can see the prokaryotic structure or prokaryotic cell. Here you can see that in the middle there is something which is called nucleoid. This is the genetic information. It could be DNA as well as RNA, because you have DNA single cell organisms as well as DNA organisms. So, for example, everyone is familiar with COVID. So COVID is a virus. Some scientists believe that also virus is also an organism, it's also a living matter. Some of them, they really think that viruses are not alive. But just let this discussion to scientists, and we can talk that, for example, coronavirus as well as the flu virus, they are the DNA viruses. They are the RNA viruses, sorry, they are the RNA viruses. But another sort of pathogens, usually they are also the DNA viruses. What it means? DNA virus, RNA virus. So RNA virus mean that the genetic information is present in the nucleic acid, which we call RNA, ribonucleic acid. If it is a DNA virus, it means that the genetic information is present in deoxyribonucleic acid, so DNA. There is a difference also. There is no envelope of, but this one, it's not a virus. Yeah? It's just a cell. It's some bacteria, prokaryotic bacteria. It's more complicated. This is a little bit higher level than virus. But as you can see, there is no envelope, so no nucleus. So it means that the DNA or RNA, but DNA is better. They are much more common, the DNA organisms. It's present there without any envelope, without any protection, without any special coiling. And also the DNA, which is there, is circular. So it means it's like it has no end and no beginning. It's like Ouroboros, something with head and tail connected together. So it's a circular DNA. This is the genetic information for the prokaryotic cells. Next time we will have also some lectures for nucleic acid. And we are going to talk about the DNA and RNA structure more deeply. But just now remember that in prokaryotic organisms, the DNA, the genetic material, is without any special coiling, without any special envelope, and it's circular. It has no beginning and no end. What else is there? These blue dots, it's ribosomes. This is the factories for the preparation of proteins. There is also the cell membrane. This is just the first protection. The cell membrane consists from special chemicals, which we call it's fat as well as phosphate. It's phospholipids. It's a double layer, and on the surface it's charged. In the middle, it's very hard to dissolve anything which is on the liquid base, like a polar molecule, like a water, because there are the tails coming from the fatty acids. And also for the majority of prokaryotes, we can find there some cell wall. Usually cell wall is created from some stable chemicals like chitin, and also from starch sometimes, and cellulose, and so on. Usually there is another envelope which is called capsule. So it's something which protects the single cell organism. And sometimes there are some trichomes, or here some flagellum. So it means something which allows some limited movements for these organisms. So prokaryotes are very simple organisms, evolutionarily older, and also the number of organelles are limited. We are going to focus more on a eukaryotic cell, because eukaryotic cell, it's a type of the cell which is present in human bodies, but not only in human bodies, but in all animal bodies, as well as higher plants bodies. Here in a eukaryotic cell, you can see nucleus. This is the very important organelle. In the middle of nucleus, there is nucleolus. So it means there are two envelopes, and in nucleus there is present the DNA, the genetic information. This genetic information is not circular, it's really linear. Usually it's present in form of chromosomes. So for example, in humans we have 23 pairs of chromosomes, so 46. And the DNA, which is present in chromosomes in our DNA, it's not circular as is usual for prokaryotes, but it's linear. There's the beginning and the end. And it's not present like lying or floating in some liquid, but it's coiled on some proteins and protected by this way. But anyway, we are going to explain it more deeper in next lectures. Another organelles which are in close distance to nucleus, it's endoplasmatic reticulum. We have two types of endoplasmatic reticulum, rough and smooth. And here blue dots, again, the ribosomes, the factories for proteins. And these are the mitochondria, the red organelle in this case. And we also call this organelle as a semi-autonomous organelle. It's very, very interesting and very important organelle. It's actually it's the factory for electricity of every cell. There is a difference between the plant cell, plant eukaryotic cell, and animal eukaryotic cell. Animals, there is no cell wall, so we have only plasma membrane. On the other hand, plants, they have plasma membrane and also the cell membrane. So this is some basic difference among these two types of cells. So let's talk about nucleus. The main function of nucleus is just to separate DNA, the genetic information, from the rest of the cell, from the cytoplasm. Also, the function is protection. So it means that, as I mentioned in the first lecture, that when the life was created, there were a big radiation. And you probably know that DNA can be damaged by strong radiation. So therefore, some protection is essential. So there were protection coming from the water, protection coming from the plasma membrane or cell wall, and also the second protection coming from the nucleus and nucleus membrane. So these three-walled protection helps to keep DNA just protected or aware to contact with X-ray radiation or UV radiation in general. Close to nucleus, there is endoplasmatic reticulum. You need it's this part, this blue part with the red dots. So it means it's a roof endoplasmatic reticulum. Here you can see two endoplasmatic reticules, the red one with the red dots. So it's a roof endoplasmatic reticulum and the smooth endoplasmatic reticulum. So the root endoplasmatic reticulum is really neighboring structure of this nucleus. And the reason is that endoplasmatic reticulum is important for the proteosynthesis. We have a special lecture for proteosynthesis. So it means, what means proteosynthesis? We have DNA in nucleus. So DNA never left, it's never leaving nucleus. It's always sitting inside. However, it's a book for cooking of proteins. So it's written, the structure, the primal structure of all proteins is written in our genes, in our DNA. So therefore, we have some special mechanism which serves for rewriting, transcription of information present in DNA to messenger RNA. This molecule is leaving nucleus and it gets outside to cytoplasm, to ribosomes, these dots which are in close distance or sitting on the surface of root endoplasmatic reticulum. And there they serve as a template for the synthesis of primal structure of proteins, of peptides. Later on, this primal structure of proteins, peptides, goes in the middle to the vesicles of endoplasmatic reticulum. There it's modified chemically. The secondary, tertiary, and quaternary structure of proteins is provided in root endoplasmatic reticulum. Then it's packaged, sorted, and transported to some fine place of proteins utilization. So this is the main task of endoplasmatic reticulums, roof endoplasmatic reticulum. Smooth endoplasmatic reticulum. This is also a sort of or couple of vesicles. They serve for the storage of calcium ions. Probably you know from the muscle contraction, the sarcoplasmatic reticulum. So it's something similar. The structure is very the same one because smooth endoplasmatic reticulum serves for the regulation of the calcium ions release. And by this way, it also controls the opening of some channels. And we know it practically, the most famous role for calcium is the control for the muscle contraction. We are also going to talk about it. We have a special lecture for muscle contraction. And also the second task for smooth endoplasmatic reticulum is the metabolism or synthesis of lipids. If we go a little bit further, we are getting to ribosomes. Ribosomes are, there are many ribosomes within every, especially in the synthetic phase of every cell. There are thousands of ribosomes, and every ribosome is the single factory for the proteosynthesis of the They serve for the translation of the information from messenger RNA to the structure for proteins. The ribosomes has two parts, the big subunit of ribosomes and the small subunit of ribosomes. There are some special tools, so places in this unit, and every place has a special reason and special task what to do during the proteosynthesis. Again, we are going to talk about it in a special lecture, which is dedicated to proteosynthesis. So your presentation should be quite general. Just describe what is important. So you have 6 minutes together to one organelle. So every student is going to speak only 2 minutes. So just try to measure it. So you can say just very rough and very highly important information, the most important one. It's very important to do this exercise because you are going to remember the most important information, because you are going to distinguish which information are more important and which are less important. So this exercise, it's highly important for your future education process. You can apply this way of learning also to another subjects, not just for this one. So Golgi apparatus. Golgi apparatus, it's again another organelle. It plays a very important role, especially in apoptosis, because it's in close cooperation with mitochondria, because mitochondria, they are also related with apoptosis. Also, it plays a very important role in packaging or assorting of proteins, which are synthesized during the photosynthesis. And also it's using for packaging and assorting for lipids. So it means that the material like proteins and lipids are getting through the Golgi apparatus membrane. It goes to vesicles, and some membrane or some envelope, some wrap is made on the group of these chemicals. By this way, these vesicles are used for the transport of proteins and lipids to some target places within whole organisms. Also, some chemical reactions are provided in Golgi. Namely, it is glycosylation. So it means if I have a protein, usually it's quite rare to have only proteins which are made of amino acids. Usually this amino acids protein, it's we are adding there some another chemicals like sugars, lipids, phosphates, and so on. And if we add sugars and phosphates, we call it glycosylation. If we add sugars and phosphorylation, if we add phosphates, and these two chemical reactions are provided also in Golgi. That's the most important reactions which are occur there. And finally, mitochondria. This is very special organelle because it seems to be like an organism. Indeed, in the past, scientists, they expect that mitochondria were the prokaryotic organisms. So it means that it is expected that mitochondria was eaten by a eukaryotic cell. The reasons why scientists have these expectations there are several reasons why. First of all, mitochondria has own DNA, and this DNA is circular, so it means it's a prokaryotic DNA. So we have also prokaryotic DNA in our body, and it's mitochondrial DNA. So it has no beginning and no end. Okay, there are also mitochondria is important. So it means that this organelle was eaten, phagocytated by organism, by eukaryotic cell. However, it wasn't digested. So it means that the organelle remain within the cell inside without digestion. So therefore we call it semi-autonomous organelle. In the DNA, which is in mitochondria, the majority of the genes which are there, they serves for the synthesis, or encoded there are the proteins, which are enzymes used for digestion, for metabolism. So mitochondria, they serve as factories for electricity. So the cellular breathing has occurred inside mitochondria. Cellular breathing, so it means oxidation. So it's something like that if we are going to burn something, just you can take a candle, and if you are going to candle, surprisingly, I have a candle in my office. So if I just light on the candle, there will be a fire. And if I put there a paper, I'm going to burn it, naturally. But chemically, the process which we call burning, it's called oxidation. And exactly the same one happened in mitochondria. So try to imagine that how big mass will be in my office if I'm going to burn something on the floor. Terrible. It will be absolutely non-acceptable big mass. And try to imagine what happens if you are going to make a burn inside your cells. So you are going to kill your cells. You are going to kill yourself. Therefore, we have mitochondria. So mitochondria has a special protection. So there are two membranes. The outer membrane, this one, the smoother one, and this membrane which is in the middle. This middle membrane, it creates some cristae. So it means that the surface which is inside is increased, enormously increased, because the presence of this cristae. And actually these cristae are creating some chambers or rooms or little reactors which can be used for the burning of substrate, in the meaning of sugars, lipids, or even proteins, just to be burned or oxidized, and the energy from these can be used and stored in the form of ATP and then used for another processes. So, is it clear? Questions? So let's check. So all the students are already written. Nice. So my question is, Natalie, Tara, and Gaone, you are going to cooperate together?
Yes, we will.
Perfect. So everyone from this group agree?
Yes.
Perfect. What about Yorgos, Jakub, and Behnut? Are you
Yes.
going to? Okay.
Yes.
Yes. Fine. What about group number 3? Hossein, Albara, and Hassan?
Yes.
Mm-hmm.
Yes.
Yes. So Muhammad, Part, Aklara.
Yeah.
Perfect. Ivan, Muat, and Sanskurt, Sunny.
Yes.
Alden, Daniel, and Zani.
Yes.
Faisal, Marcos, and Alexandros?
Yes.
Yes.
Perfect.
Yes.
So be ready for next lecture. We will start from 1 and try to prepare. My recommendation is to prepare a PowerPoint presentation. You are going to share the screen as I do it, and you can switch. So first will be, for example, for endoplasmatic reticulum, for smooth endoplasmatic reticulum, first speaker will be Natalie, second Tara, and the third one Gaone. So in the order you are written here, you have to keep the same order. Every student is going to speak just 2 minutes, not more, 2 minutes. You have to prepare together the presentation, and you have to train if you are able to fit this 2-minute limit. So together, 6 minutes. Depending on your on the quality of your presentation, you will receive some points. I'm going to evaluate how you cooperate and how you prepare the meaningful presentation. So also, if you are able to recognize the most important information from the less important information. This is the first. I'm going to evaluate also the quality of the presentation. So the pictures, yeah, the design, the visibility. Please do not put there too much text and do not read it. This is something, this doesn't work. Just to imagine that I'm going to teach you by this type of presentation, full of text and going to read it. It doesn't work. It's boring, and this is something which is not useful. Do you agree? So do
Agreed.
Yes, but 1/3 of students present like this way. So therefore I'm mentioning this one. And the last thing, I'm going to evaluate also the level of your speech. So try to make a sense, use simple sentence and try to intonate, try to put some accent to important words, to keywords. Try make it simple. This is the recipe how to succeed it. This is also the training for you, because you can be an expert, you may have many knowledge, many experiences, but if you are not able to present it, who takes care? Okay, that's clear. I'm going to leave you because now you have something like 25 minutes. Try to prepare for your presentation next time. So you have something like 25 minutes to make some task, to aims. And next week you can work on that. I'm going to prepare the test, and I'm going to write here to another list the score from the test. Yeah? Is it clear?
Yes.
Yeah.
Perfect. So I'm looking forward to you next week. And I am really curious, and I'm looking forward to your